Okay, questions number 7, page 50. Now, we've seen kind of like this question before, and we did have a best practice for these type of questions. So you can say you got some chickens. Gene for rooster is dominant to the single. Okay, gene for feathered leg, capital F, is dominant to clean leg, little f. Now here, it doesn't say that there's any interaction going on and giving you a different genotype, or sorry, a different phenotype when the R and the F are interacting together. So this, to me, is just another type of dihybrid. We got two traits. We got one for the comb, we got one for the legs. Okay, so let's go through that and let's see if we can come up with a legend. There's no weird interaction going on here. So it's saying gene for rose, capital R, is, as long as I have a capital R, it's going to be a rose. If I have two little r's, that's going to be a single. Now it's saying gene for feathered leg, capital F, doesn't matter what the other allele is, it's going to be feathered. And if is dominant to clean legs, so if I have clean legs with no feathers on them, it's going to be little f's. Okay, and that's going to be clean. So no interaction, nowhere does it say that one gene is interacting with another one to give you a different type of phenotype when they two interact. So just a regular kind of dihybrid put into all these little polygenic questions. Okay, now this is the strategy. We have a rooster A that's hooking up with C and a rooster A that's hooking up with a hen D. Okay? So when you have multiple matings going on, remember we said that the key strategy here is to use a table to sort this out. And they're asking you to determine the genotypes of the parents. So you know when you have to look at the genotype for the parents, you can't be doing Punnett squares, because that's what you have to put in, is the genotypes isolated into gametes in a Punnett square. But you can't do that here. So we're going to do a table. So we got a lot of, it's like Jerry Springer. We have we have roosters hooking up with hens, and we got other roosters in the action there as well. So to sort this mess out, let's do a table. So we're going to go rooster A. Okay. Next one we have is hen C. We also have a hen D. And we also have a, we already have a hen C, but we have a rooster B. Okay, do we have any other individuals mating here? No, that's enough. So let's do a table. Again, best practices. We've seen this question before when we were doing just regular dihybrids. Now here it says all, and it's bolded, all 4 birds are feathered leg with rose comb. So all of these 4 birds are feathered. So if they're feathered, they must have a capital F. We don't know what their other allele is at this point. All of them. Because it's saying they're all feathered. So the strategy is to first do a table, put what you do know. They all have to have capital F's because they are feathered, but we don't know the other allele. We don't know if they're homozygous or heterozygous feathered. And they all have rows. Same thing with that. If they're all rows, have to have a capital R. And again, we don't know what the other allele is, but they all have to have capital R's. So put what we do know, put a line indicating that we don't know what that is. Now we're going to go through the mates, or the pairing, and see what kind of offspring they produce. And of course what we're looking for in the offspring is not just any old offspring, but recessive offspring. We love recessive offspring. So it's saying rooster A and C are hooking up. Okay? And they produce some offspring. Doesn't say how many, but we don't care. All of them are feathered. And most, but not all, when it says most are rows, that means we have to assume that some were single. Otherwise it'd say all are rows. But they didn't. They said most. So they did have some single. Now all of them were feathered. So if they're all feathered, again, you got to do this with a pencil. If they're all feathered, we're going to assume at this point that they're all capital S. Because every time they made it, all their offspring had feathered. But because they had some single, we know that both these guys must be carrying a recessive allele R for the single trait. That's how they got some offspring for single. Okay, agree with that? Now this might change, that's why we need a pencil, because we also know that rooster A hooked up with hen D, and they had some offspring. Okay, and they said, again, all we're circling in this is the recessive trait. They had feathered, but look at this, they had some clean. And clean, if we look at our genotype, 2 Fs. So now we have to go back and say, whoops, we got some more information on this rooster A. He must be carrying a little recessive allele for clean, little f. Okay, and also this D must have a little f as well, because that's the mating we were looking at there. And all of them were rows though. So it's saying all of them are rows. Now we don't go back and change this little r on this rooster A, because when he hooked up with C, they did have some single. But if they are all rows, we're going to assume that this guy's a capital R. Because as long as you have 2 capital R's, homozygous, those guys are always going to produce rows. Okay, now again, use a pencil, we might change this. Rooster B and C, these guys hooked up. Okay, and it says, again, all we care about is the recessive traits in the offspring. They produce some birds, some of them feathered, but some of them clean. So if they are clean, that means those clean individuals had little f's, one came from each parent. So we know this is a little f, and we have to go back here and get rid of that, because now we found some more information, and we know that that must be a recessive allele for clean, little f. And had rows, most of them are rows, but did have some singles, combs. So if they had single combs, we now know that this guy must be a recessive allele for single, for them to ever have any single offspring. So you did have to, because they made it, there was some other mating, we got some more information, we had to go back and change a couple of those genotypes. But in the end, we got it all filled out. And the only one that's different is this guy right here. Okay, so again, best practices, do a table when there's multiple mates going on. Put what you do know about their genotype, align what you don't know. Look for offspring, but not just any old offspring. You're looking for recessive offspring to start filling in the lines until eventually you come up with the genotype of all the parents. Okay, any issues, give me an email. Thanks.